Hi, everybody. I'm Andrew Lowe, and on behalf of the MIT Sloan School of Management, the Laboratory for Financial Engineering, and the Golub Center for Finance and Policy, I'd like to welcome all of you to this very special event with our distinguished speaker, Stéphane Bancel from Moderna. Before I get started today, I want to just say a few words of thanks to various individuals and organizations that have helped to make this webinar possible. I want to first start by thanking Moderna, and in addition to Stephane, also Lavina Taluktar, for making this possible. Here at the LFE, Crystal Myler was involved in the logistics, and Deirdre Wade at the Golub Center. And thanks to all of you for joining. And actually, I have to add a personal thanks to Cutting Edge Systems, a home automation firm that I use to help set up my home network. We had a big internet outage last week, and they came right away and helped to restore it. So the fact that I'm able to be here with you today is due to Cutting Edge, a fantastic firm. I also want to thank Verizon for sending out a worker during this very difficult time. All of these essential workers are really taking risks in order to help the rest of us. So thank you all for doing that. Now, I want to start with a little bit of background, if you don't mind, because I'd like to provide a little bit of context for why it is that the Laboratory for Financial Engineering and the Golub Center for Finance and Policy would be hosting an event with Stéphane Bancel at Moderna. What is our connection to the world of vaccines and biomedicine? Well, let me first start by sharing my screen, because I'm going to show you just a couple of things and then turn it over to Stephane. But I want to set up what he's about to show us with a little bit of background on exactly what it is that we're looking for from the perspective of an economic analysis of these issues. So I want to start with the fact that finance and biomedicine are inextricably linked, because basically you need money to develop drugs and devices. And so the financing is actually quite relevant. And part of the challenge of financing biomedical innovation is the underlying risks that are involved with these kinds of efforts. The fact that you really need to deal with these kind of binary risks really requires a different approach. So recently, we launched the initiative at MIT called Project Alpha, Analytics for Life Sciences and Professionals and Healthcare Advocates. The idea behind this initiative was to be able to develop better analytics to assess the risk and reward of drug development, particularly with regard to the probability of success of different clinical trials. So if you look at this chart, this is the most recent estimate that we produced, thanks to data from our collaborator Informa. We used hundreds of thousands of clinical trial events across various different diseases and drug development pathways to estimate the likelihood of success of different clinical trials in different disease areas. And so I want you to pay attention on this graph to the orange bars, which is the probability of success from phase 1 all the way to approval for drugs in different disease categories. So on the very left, you can see that oncology has probably the lowest success rate. Historically, it's been a little bit less than 5%. So less than 5% chance that when you start off with a compound at a preclinical stage, you can get it through phase 1, phase 2, phase 3, all the way to approval. Now, if you go to the right of this graph, you'll notice that vaccines has a probability of success of 40%. Among all of the different drug indication pairs, vaccines has by far the highest rate of success. So naturally, you would think, no problem. We ought to have lots and lots of vaccines out there for all of the different infectious diseases that are a threat to us. So it was a real surprise when my co-authors and I, Chihim Wang and Qinwei Xia, when we did the analysis, we actually found that even though the probability of success is quite high, and in a recent paper that we're about to release, we're going to show you even more refined measures of probability of success for different kinds of infectious diseases and different kinds of therapeutics. What was surprising to us is that there were fewer and fewer companies involved in developing vaccines. In fact, right now, there are only four big pharma companies that are focused on it. A number of them have left the space, and smaller companies have gone bankrupt and are really not developing vaccines anymore. So this was a bit of a surprise. And in a paper that we recently posted to medRxiv, so you can take a look at it at your leisure. My co-authors, Jonathan Vu, Ben Kaplan, Shomesh Chowdhury, and Monique Mansour, and we tried to understand why it was the case that on the one hand, the probability of success was so high, and on the other hand, there were fewer and fewer vaccine developers and vaccines out there. So we did a simple business simulation to ask the question, what kinds of rates of return might investors earn for investing in vaccines? And what we found was really shocking. If you take a look at the vaccines that are on the hit list of the Coalition for Epidemic Preparedness and Innovation, the CEPI organization, you'll notice that in table 1 of our paper, we do a simple back of the envelope calculation to ask the question, what's the expected annual revenues in case you're successful? Because you only get paid typically when there's an outbreak, and the probability of an outbreak varies across different infectious diseases. And what we found really shocked us. The typical expected annual revenues from these vaccines is generally pretty low, on the order of single-digit millions of dollars. Compare that to a cancer drug, which, if approved, can bring you revenues of billions of dollars, or in some cases, tens of billions of dollars annually. So from the perspective of purely economic considerations, it doesn't really look like that great an investment. And when we calculated the IRR, the internal rate of return of a portfolio of these vaccines, what we found is that given the pricing models that are out there right now, we're looking at rates of return of around negative 60%. Now, obviously, that's a crude generalization because we're using relatively broad numbers, but even a more refined simulation that we've been working on does not really seem to help. The economic model for vaccines is broken, and that's one of the reasons why, thankfully, a number of nonprofit organizations have emerged to deal with this issue, including CEPI, the World Health Organization, Wellcome Trust, CARB-X, the Gates Foundation. They've done an admirable job of doing what they can, but it's not enough. What we need is to get the private sector engaged, and so we need better models. Now, one of the silver linings, if there is any, of COVID-19 is that it will raise awareness of this issue, and it will bring more attention and, more importantly, more resources and more of a commitment both from the public and private sectors. And that's one of the reasons why we're hosting this event with Stéphane Bancel. So without further ado, I'd like to turn it over to him. Stéphane Bancel is CEO of Moderna, the company that had the very first clinical trial for COVID-19. He's going to tell you the story. It's really amazing. From the first time that we had the genetic sequence for the virus to the time that they dosed a patient, 63 days, an astonishing story. And so we're really delighted to have him with us. So please join me in welcoming Stéphane Bancel from Moderna. Stéphane?
I think you raise a good point around vaccine and the list you had. I think it's important, and maybe we can talk about that in the Q&A, to understand that there are two types of vaccines. I think vaccines against pandemic threats, like the Marburg and the Zika that you had on that list. And indeed, because you don't know if you're going to be able to sell those products, it is really hard to have a business model against it. And that's why you need public-private partnership. But I think it's important also to note that there are still many, many viruses that cause a lot of harm in this country that do not have a vaccine yet today. We're going to talk, of course, about COVID today, but there are a lot of viruses like CMV, cytomegalovirus, RSV, where you have thousands of Americans dying each year and no vaccines on the market yet. And we believe those are multi-billions product opportunities. So you have already this dichotomy in the vaccine world. So thanks for having me. I've been a big fan of your work. I've been in this industry for 25 years, and I've been always frustrated that most of the analysis done on probability of technical success for infectious disease were bundled together, where people will look at antibiotics, antiviral treatments, and infectious disease vaccine all bundled together. And for me, it never made any sense because those products are very different from their mechanism of action. And I was very thrilled a few years ago when I started to see your data on infectious disease vaccines, so that people can realize the very high probability, like you've shown, of a vaccine once you get good phase 1 data to get to approval. So let me start by making a few comments. Before that, I need to remind you that investing in Moderna, given we're a public company, entails quite a number of risks. Those risks are detailed on this slide, but this is available on our website and on the SEC website. And also that I will be making forward-looking statements that might change. As you can understand, the situation around COVID is extremely fluid, from our understanding of a virus, our understanding of a disease. And so we're working in real time towards our plans to get this vaccine, hopefully, if it works, approved. And so we might change our plans as early as a few days from now, based on what we learn in our discussions with the FDA. So with that, let me spend a few slides just to explain what we do at Moderna for those of you that are not familiar, and then go into COVID-19 vaccine. So we started the company 10 years ago based on this idea, which at the time many of us, including myself when I first heard the idea, was to use messenger RNA to inject messenger RNA into humans so that you make your own protein on demand. This was a pretty bold idea at the time. We had all been trained believing that mRNA is a very unstable molecule, that it's highly immunogenic, and so it will never be a good molecule to make medicines. And mRNA, as you know, is the central dogma of biology, is a molecule sitting between DNA and protein. It's the information of life. We call it the software of life. It carries instructions from DNA to cells to make protein. There are 4 reasons why, once we started to think about this idea of starting this company around messenger RNA, I got extremely excited. Having trained in big pharmaceutical companies, having done small molecule, large molecule medicines, there are 4 things that got me very excited from the first moment I started to think about this company. The first one is the very large product opportunity. If you think about it, the biotech industry over the last 40 years has created amazing medicines, created a lot of value on the way by focusing on secreted protein that are made somewhere in your body, like insulin in your pancreas, and secreted into your serum. But if you look at human DNA, around 2/3 of the protein encoded in our DNA code for transmembrane protein that you can see, if you can see my little mouse moving on the membrane of a cell, or intracellular proteins. And so 2/3 of a protein into the human genome are not really addressable using recombinant. So that's a gigantic market, obviously, as you can imagine. The second piece that got me very excited, because over the years I got very frustrated of a failure of drugs in clinical trial, is we believe that the probability of technical success of our mRNA medicine should be materially higher than reported probability of technical success of traditional medicine. And we thought there was 2 reasons for that. The first one is mRNA is a platform. The way we make mRNA for one vaccine is exactly the same way we make mRNA for another vaccine. The chemistry we use to make the mRNA molecule is the same. Manufacturing process is the same. Lipid that we use to formulate and package the mRNA in the vial is the same. Manufacturing process is the same. And so the way you think about it is because mRNA is an information molecule, the difference between our flu vaccine and our Zika vaccine and our COVID-19 vaccine is only the order of a nucleotide, the 4 letters of life on the message. And so it made a lot of scientific sense that if one vaccine was going to work in the clinic, the next vaccine, if you are coding correctly for the sequence of a virus, should have a very high chance of working. And the second piece that I liked a lot about this technology is that we do not invent biology. And what I've learned in the last 25 years in this industry is fighting biology is never a good idea. And so if you think about the raw material we use at Moderna, we use either human DNA and the instruction of a gene or of multiple genes to code into a cassette for mRNA molecule, or we use the sequence of a virus and we pick a protein or several proteins of a virus that we literally drop into our cassette and we're ready to go with a new drug. So these platform features that exist in the tech world, but that not really existed in the biotech world or in the pharmaceutical world with small molecule, made it extremely exciting. The third piece is speed. Because it's a platform, because we use the same manufacturing process for everything, we will not have to wait for engineers to invent how to make that one drug at a time. It's the same process for everything. And so we thought that if we digitalized and robotized and industrialized the company correctly early by doing those investments, like any platform, then we could scale the company very quickly. And last but not least, what is very exciting about messenger RNA is it is made in water with enzymes. And so it's a cell-free manufacturing process, so massively smaller and thus cheaper than using recombinant. Plus, because we make all the products in the same plants, we do not have to build, during the phase 3 study of a drug, a plant dedicated for that product. We use the same plant to make everything. So we don't have any capital at risk as we await the phase 3 data. I was traumatized a few times being in big pharma when the drug fails in phase 3 and you have to go explain your board and your investors that you have to write off a $200 million or $500 million factory. It's never a happy day. And so the cost of goods also should be very interesting. So because we believe this could be a new class of medicine, we said picking one drug and going to the clinic and hoping every day that it's going to work made no sense. Because we thought this would be a new entire big class of medicine, being able to deliver potentially dozens and dozens and dozens of drugs to the market over a long period of time, we said we cannot manage the technology risk of the unknown of the technology. So we said we cannot do one thing. We need to do 6 different technologies in parallel. And it's what you see on this pictogram. We've tried in the clinic in parallel 6 different applications of a technology, different route of administration. On the far left, the vaccine are intramuscular, like a typical vaccine. On the far right, the product in orange are injected IV, intravenously, to the patients. And they use different formulation systems. So there is no correlation in terms of a technology risk of the orange product, of a green product, of a blue product. And to manage biology risk, which always exists because some diseases are well understood, some diseases are poorly understood. Think about Alzheimer, think about a virus like HIV. 40 years after the discovery of HIV, we still do not have a vaccines for it. And it is not for many, many people spending their entire life trying to make a vaccine work. The biology of HIV is extremely complicated. And so we try to build for each of those applications a portfolio of several drugs to diversify, like you build a portfolio of stock to diversify the biology risk. So that's what we did. And so we took so far 16 different drugs into the clinic, and we said we're going to listen to the clinical data to tell us where the technology works, where it works. We'll do many more of those type of products because mRNA is software, it's information. You can copy and paste and do a lot of drugs with same technology. Things that will not work, we will shut it down and move to other things. 2019 was a very important inflection year for the company because 2 of our applications, the prophylactic vaccines for infectious disease and systemic secreted and cell surface therapeutics that you see here in pink, got extremely exciting clinical data. And so what we announced in January of this year is that those 2 modalities, those 2 applications, we believe have been massively de-risked from a technology standpoint. And so we said, we're going to invest many, many more products in those 2 things. And in the first 2 months of the year, we announced already 5 new products moving from the labs into the clinic in just 60 days. And for the 4 exploratory modalities that you see here on the matrix on the right, we are being very disciplined. We are waiting for the clinical data. Most of our products, except the one in orange, are already in the clinic. The VEGF in purple is in phase 2 with AstraZeneca. The personalized cancer vaccine in dark blue on the left of the matrix is also in phase 2 with Merck combined with Keytruda, their oncology drug. So the way you want to think about the company now is we have those core modalities where our strategy is to invest a lot of capital to get a lot of drugs going because we believe the technology risk is massively de-risked. And on the right, the exploratory modalities where we are still waiting for clinical readout to decide, do we move them in core modalities and do more of those, or do we stop investing in those products? What is exciting about Moderna, and I will need more time to talk about it, so it won't be for today, is we believe there are many more modalities we can create over time. One that is not on this slide yet because it's still in the labs, but it's public information, so I can talk about it, is we're working with Vertex on trying to deliver messenger RNA via aerosol via the mouth and the nose into the lungs for cystic fibrosis kids. So it is still in the labs, but we're making nice progress there. So if I look at our vaccines today, I won't go through the whole list. You will see the SARS-CoV-2 vaccine at the bottom here. Those are all the vaccines that we have in development. So this is not research, this is development. Most of those products are in the clinic. And the CMV at the top, we announced recently that we finished enrolling all the healthy participants of its phase 2. And we are working hard to start our phase 3 as early as next year. So we have had positive data in 6 different vaccines so far. So why do we love messenger RNA for infectious disease vaccine? The punchline is that it mimics a natural infection without giving the humans the virus. So if you think about it, what happened into a natural infection, let's say with corona? You get the virus into the human body. It is an mRNA virus. It goes into human cells and hijacks your ribosome to make protein to replicate. And it keeps doing that and replicates. And when you have a very, very high number of copies of a virus, you get disease. What do we do with messenger RNA? We pick one or several proteins that we think are very interesting and important to teach your immune system how to mount an antibody response so that if later on you get natural infection, the antibody made by your immune system will bind to the virus, neutralize the virus, so it cannot replicate. And so if you think about it, it is the most adapted technology to go after infectious disease vaccines because we use the human cells to make the virus protein exactly like if you had the natural infection. We just do not give you the virus. We've done it many, many times. We made multiple mRNAs in a single vial. We've shown we can go very fast, and we use the same facilities, allowing us to move very, very quickly and not to take capital risk for risky projects. So what do we know so far of our technology? We have dosed more than 1,000 healthy subjects across different studies, up to 400 micrograms. The vaccines have been well tolerated. The profile looks very similar to marketed adjuvant vaccines. And so far, the 9 vaccines we put in the clinic, we had positive data already on 6 of them. So you can see over the summary here, I won't go through everything, just 3 vaccines that we announced in February as new vaccines, including mRNA 1273, which is a SARS-CoV-2 vaccine. So what I want to share with you is just one set of data, but I think it's important. It's data that we published in our S3 in February. This is the preclinical data for a related coronavirus, which is MERS, the Middle East Respiratory Syndrome that many of you will recall happened in the Middle East in Saudi a few years ago, and then a few years later again in South Korea. So what you see here is a rabbit model. On the left panel, you see injection over time and the concentration of the antibody in the blood. At the bottom, what's stayed at 0 in light gray is, of course, the placebo group. You would expect the placebos, when you give them water, the rabbit obviously do not make antibodies against MERS. So this is all as expected. What you see in black is the antibody levels after 1 dose. So you see you mount an immune response around 10 to the 2 of antibody titer. And then in the dark gray, you see a 2 dose. So you see at day 21, we basically inject the vaccine again as a boost. And you can see the black and the dark gray line are on top of each other from day 0 to day 20, which is not surprising because both animal groups have only 1 dose of a vaccine. And then at day 21, you give another dose, again intramuscular, and you see how the level of antibody goes up by a log. and is sustained at that level over time. And what you see is at day 42, we give a very high dose of a virus in what is called a challenge model to the animals. What you see on the right panel is the viral load, which is how many copies of a virus can you find in the animal. And you see again in light gray, the placebo group, in black, the animals with 1 dose, and in dark gray, the animal that got 2 doses. And you see the result in the throat, in the nose, and in the bronchovehicle veolar lavage. Basically, you put a lavage into the lungs to understand the viral load into the lung. And what you can see is that in the throat and the nose, you almost at baseline, meaning you cannot find much virus after the vaccination. You see a little bit in the lung, which is very typical of vaccination, because in those models, you give extremely high dose of a virus, those that most of the times are way higher than what you will get for a natural infection. And what you can see in the nose, we can reduce by 3 logs, and in the lungs, we can reduce by around 4 logs. So we are very excited to see this data that was realized with the NIH, with a team of Dr. Tony Fauci. So let me turn now to what happened with the SARS-CoV-2 project. At Moderna, because of our mission of infectious disease, we are always looking at new outbreaks. And in the first days of January, before even the world knew this was a corona, we were starting to monitor the outbreak, talking almost on a daily basis with Dr. Tony Fauci's team. And a few days into January, we learned it was a corona, not a flu. Everybody was initially thinking it was a flu because there's been a lot of flu outbreak in China in the last 10, 20 years. And on January 11, the sequence of the SARS-CoV-2 virus was published online. Our scientists working with the NIAID team studied the sequence of the virus, realized that the spike of the S protein of the virus was extremely close to the one of the MERS virus. Because of the good data they had done, and I only showed you one data set, they've done much more analysis over the last 18 months, 2 years, working on the MERS. The team decided to code a vaccine for the spike S protein. That vaccine design was locked down on January the 13th. By February 7th, our team at our plant in Massachusetts had already done and finished the vials for the vaccine for the clinical study. So as you can see, less than a month, that night, we started doing all the quality control tests, including sterility testing. By the 21st of February, the product passed the test. We sent a certificate of analysis to the FDA. That was the last piece missing in the IND file that we sent to FDA to ask the FDA the authorization to start the clinical study. We submitted, of course, all the manufacturing file. We submitted, of course, preclinical data that we had done in the previous few weeks in the labs. And we shipped the vials once the certificate of analysis was complete and successful to the NIH on February 24th. The FDA opened the IND with giving us and the NIH a green light to start the phase 1 study on Monday, March 2nd. And as you know, it has been widely publicized. On Monday, March 16, the first subjects in Seattle were dosed with a product. I'll comment later if there's a question around it on how could we go so fast. I want to keep moving for a couple of slides before taking questions. So if you look at Moderna today, the company has a very broad pipeline, more than 20 products on the pipeline. We're preparing our first phase 3. We are making the phase 3 material for the CMV product as we speak. We have 4 products that are either in phase 2 or preparing for phase 2, 12 ongoing phase 1, and already 10 phase 1 that have positive results of safety and a sense for activity in the clinic. So 6 vaccines, as we discussed, but also a personalized cancer vaccine was featured last year at ASCO, the big oncology conference in Chicago. Our OX40 ligand product has shown that we can make the protein in humans that we proved through biopsy of human tumor. AstraZeneca has published a very interesting paper, which is on our website in Nature, showing that they can increase blood flow in human by injecting VEGF protein. VEGF is a protein that basically induces the formation of new blood vessel. And we showed our first antibody product that is 2 mRNA in every vial to make a full functional IgG, which was funded by DARPA from the Department of Defense to get an anti-chikungunya human antibody. And that data was presented last year. So if you look at it by therapeutic area, we today have 7 vaccines for major unmet needs. And I think it is very important to understand this in the strength and the diversification of the Moderna portfolio. In vaccine, or 7 vaccines are going after viruses for which there is no vaccine on the market. And some of them, as we discussed, are causing thousands of deaths in the US every year, much more obviously, as you can imagine, around the world. We have 5 cancer drugs that are immuno-oncology, that are all in clinical trial as we speak, 5 rare disease programs, 2 autoimmune disease programs. More than 1,700 healthy volunteers and patients have been enrolled in our studies. The team still is growing very rapidly. We had more than 800 people at the beginning of the year. We have a big plant in Massachusetts, important partnership with Merck, AstraZeneca, Vertex on the for-profit side. On the not-for-profit side, we had partnership with BARDA, which is part of HHS for Zika vaccine, which is one of those partnerships that Andrew talked about, where the government is funding a product for a very important need, because we believe Zika is going to come back. Nobody knows when, but it's going to come back. And the world needs a vaccine for when it comes back, so we don't have the human catastrophe that we had in Latin America a few years ago. Also Department of Defense, DARPA, but also the Gates Foundation. The company is well capitalized. We had more than $1.77 billion of cash or cash equivalent on our balance sheet. The company has no debt. And we have up to $183 million of grants that we can draw down as we do the work. But what is more exciting for us is what we can do for patients over time. Our team is very mission driven. We believe we have to make this technology work. I always tell the new members of the team at Moderna, the most important words in that mission statement are the second word and the last word. We want to deliver for patients. We believe this technology is a new class of medicine. We believe Moderna is just getting started. And I look really forward to the many, many years of many, many more medicines going. And especially in this time of crisis around the pandemic, we see our responsibility to be really important to move as fast as we can, while of course ensuring safety of the participants. Safety is our number one priority for vaccines because you give them to healthy people. But we want to make sure every day matters. And with this, Andrew, I'm going to conclude my remarks and be very happy to take any question you might have.
Oh, wonderful. Thank you so much, Stephane. That was terrific. And I want to start by thanking you and Moderna on behalf of really all of humanity for doing what you're doing. Obviously, it's a business, but I also know that the people at your company and at other companies are basically working round the clock 24/7 on behalf of the rest of us. So thank you for doing that.
Thank you.
I want to first start by asking Zach Whitman, who's head of AV at Sloan and who's also been terrific in helping us with the logistics. Zach, could you launch a poll to let us know who's actually on in the audience, please? We'd just like to get to know who you are. And we've already gotten a number of really interesting questions that I'm going to put forward to Stephane in a minute. But before we get to that, Stephane, I was hoping just to ask you for the current issues that are top of mind on everybody's list. How are things going with that clinical trial in Seattle? What's the time frame look like? How soon do you think we can expect that you would be able to present data to the FDA? And how quickly will the FDA be able to look at it to get to the point where we're actually able to get this to patients around the world?
Sure. So let me start by the FDA first, because I want to give them my kudos. They have been amazing. They have worked around the clock. They have responded to our phone calls and emails right away. And they have really put so much resources at the highest level at the agency to make sure that no hours is wasted in this fight against this pandemic. So they have been really wonderful, and we are lucky to have such an agency. So the phase 1 study is going very well. It's enrolling on plan, the subjects. We are hoping to get safety data, which is the primary endpoint of the study, sometime in the spring. You know, we don't control the study ourselves. This is a study that's sponsored by the NIH. And so they will be the one releasing the data. We're of course talking to them almost on a daily basis, as you can imagine. So I would say spring for safety, and then late spring, early summer for immunogenicity. What I think is important to appreciate about vaccine, which goes back to your work on the probability of success from phase 1 to approval, is you learn a tremendous amount from a scientific basis in your phase 1 in vaccine. Unlike therapeutics, where sometimes you just get a bit of a sense for safety, but you don't really know if a drug is going to be working or not. In vaccines, we basically take a blood sample from the subject who got the vaccination. We do it before vaccination, between the prime and the boost, after the boost. And what we're looking a bit like I showed you on the animal model, we're looking at how much antibody over time can you find into the blood. So you get an idea of the concentration of antibody per ml of blood, which is of course a very important information to know in terms of how much antibody are you making or not. And the second information that you get, which is critical, is are the antibody that you find in the blood neutralizing to a virus? And so basically you run an assay done into a lab, into let's say a petri dish to oversimplify, where you basically use the blood that contains the antibody from the humans, from your phase 1 study, to see can you neutralize the virus, can you stop the virus from replicating? And as you can appreciate, this is a very important thing to know, because if you have strong neutralization, it increases the probability of the vaccines to work against a natural infection that the subject will get after vaccination. If you don't have neutralization, as you can imagine, this is a very bad sign, because it means that most probably the vaccine is not going to work to prevent infection. And so this is why you see basically manufacturers or sponsors stopping their development is if they don't get safety and neutralization and a good titer level. So we're actually going to learn a lot, which is what explains the data. Very interesting your research showed, which is if you have good data on both fronts of safety, neutralization, and titer, it gives a very, very high chance of a product to be able to see through to launch.
Mm-hmm. And in terms of timing, are we still
on? So safety, yeah, we think in the spring. Immunogenicity will be late spring, early summer, because you need to wait after the second dose. You need to collect all the blood sample, and you want to do the analysis at the same time. You don't want to start testing blood samples because you want to reduce variability, of course. Mm-hmm. The variability of the assay in the lab, variability of the controls of running that assay. There's so many things that can slightly change the result, and to have a good scientific understanding of what is really happening in the clinical study, you want to basically pull all your samples and run them ideally at the same time.
Sounds good. Well, we're all waiting for those results.
So are we?
Yeah. So a question that came up from one of our viewers, Shan Shu, is that question about whether or not there's room for more than one kind of vaccine, because obviously BioNTech and CureVac are also developing mRNA vaccines. How does Moderna's approach differ from theirs? And is there room for multiple vaccines on the market?
Yes, so I think there's room for multiple players on the market. I mean, one example that everybody will appreciate is flu, seasonal flu, that I hope everybody gets, every flu season. If you don't, next winter, you really want to make sure you get it, especially if Corona comes back. We can talk about that later. And so if you look at the flu market, there are several manufacturers around the world who basically provide different variation of a product. Some products work better for healthy adults. Some products work better in the elderly setting, because the immune system of a 33-year-old, healthy adult is not the same as the immune system of a 70-year-old adult, as for example. And so in a lot of vaccine markets, you end up with several manufacturers. So that's the first piece. And what we want for the world, if I take my Moderna hat off now, is we want several vaccines to work. And so like in any problem, like you described, given positive phase 1 will get you around 42% to launch. And we don't know yet, with those new technology, the probability from, animal into phase 1 positive. This is important that there's a lot of vaccines in that race, not against each other, but against the virus, because we're all fighting the pandemic. I'm not fighting CureVac or BioNTech, I'm fighting the pandemic.
Right.
The technologies are quite different. First, in terms of the chemistry we use for the messenger RNA, and then the chemistry we use for delivering those products. Moderna has done 9 clinical studies of 9 different vaccines. More than 1,000 people have been dosed, as we discussed. So we have a very strong database and understanding of our technology. And I think the regulators as well, because of course the FDA knows all that data. To the best of my knowledge, CureVac has had 1 vaccine that they put into the clinic for an infectious disease. I think it's against rabies, if my memory is correct. And to my knowledge, BioNTech, which is more of an oncology company, has not to this day done yet an infectious disease vaccine into clinical studies.
Right. So we actually have a question from U.S. Congressman Bill Foster, who's on the call. The extraordinary human and economic damage from COVID-19 alters the traditional risk-benefit optimization of vaccine testing and deployment. In particular, this argues for earlier mass deployment of a less tested vaccine to the most at-risk population. So first part of the question, is this an analysis you agree with? Second, are U.S. regulators signaling a willingness to consider abbreviated testing protocols for COVID-19 vaccines, or should Congress step in to encourage them? And finally, are there other countries with a different risk-benefit calculus likely to adopt more accelerated vaccine rollouts? And do we have in place methods to take advantage of lessons learned from those earlier deployments?
Yes, so there's a lot of good questions to unpack here. So Congressman, thank you for your question, especially thank you for your support in all the bills that have been passed very, very quickly. As I shared, we're in discussion with BARDA, this is public information, and we really value the speed at which our politicians have moved together to help the country. So the first question on the risk reward. So obviously, given the global pandemic and the pace at which people are getting infected, as you see, it's a highly, highly contagious virus. The death toll, of course, is horrific. And unfortunately, most probably is going to get worse in the next few weeks. And as I talked about, and we can come back to it later, I am in the camp of those that are worried about next fall because of what's happening in the Southern Hemisphere already, where it's the beginning of their fall as we speak. And if you look with travel and the contamination, the R naught of this virus, I think there is a case for new outbreak in the fall that we should talk about. So the FDA is highly aware of the situation, and as I said, they have been moving extremely fast. The piece that we need to solve for is really the safety reward profile. If you think about vaccines, our phase 1 is 45 subjects. We anticipate our phase 2, which I hope could start, in the spring, late spring, will be hundreds of subjects. And the phase 3, which we are hoping to start by the end of the summer, early fall, will be thousands of subjects. Very typical of vaccine development, from dozens to hundreds to thousands. The piece that is really important about vaccine is to make sure that you don't have a safety event that happens, 1 in 1,000 person, that you will not see if you only look at a few hundred people. And then when you have an approval, you could sell to millions of people literally, and think about how people will be eager to get vaccinated very quickly because everybody is very scared of this virus. Is you could have a massive public health issue if there was a safety issue with vaccine that has not been seen and understood during clinical trial. So I believe that making sure that we go step by step is really critical. There are a lot of things we are talking to the FDA of doing, which we are not doing in normal times. And let me give you 2 examples. The first one is one that the company has taken using so far our own capital, and we hope to finalize discussions with BARDA to get help from the government, because Moderna is still losing money, every day. The company is not profitable, but it's the right thing to do for the world, which is why we've done it with our management team and our board. Is we are already making phase 2 material at risk now. We have seen no data from a phase 1, but because we cannot wait like in traditional world for phase 1 data to decide, okay, it's good, let's now invest in making the product for a phase 2, and then let's do the phase 2. We are making the phase 2 material in parallel to the phase 1 happening. So that as soon as we have not only the safety and immunogenicity, which is what you do usually, we are trying to start the phase 2 based on the safety data only. So as soon as we'll get the safety of a 3 dose of a phase 1, we do not expect to wait for the immunogenicity data, which will come a few months after, to save time, because every day matters. We will know the vaccine is safe from a phase 1 study. We will start the phase 2 study right away, and we will actually have to do it at several doses because we will not have the efficacy data from a phase 1 study. So usually what you do is you run 3 or 4 doses, in our case 3 doses in the phase 1, to understand how much neutralization you get, how much antibody titer you get per ml of blood, to pick the dose of your phase 2. And in this case, we're not going to do that. We're going to go into phase 2 with several doses so that we are well equipped to go into a phase 3 with the same idea. We are already making now phase 3 material. I was in our plant this morning to go see our team and how things were going. I wanted to be there. And they're already making the phase 3 material right now. So think about that. The phase 2 has not even started, not even getting the okay from FDA for a phase 2. We are spending capital at risk now to make the phase 3 material, so that as soon as we have phase 2 data, bang, we can go into a phase 3 and go as fast as we can. In terms of other geographies, other countries, which was a question, I think people are in the same mindset as the FDA, which is the risk-reward profile. So the other regulatory agencies I've talked to so far personally in the last few weeks want to be as aggressive as the FDA. I don't think many want to be much more aggressive than the FDA. Again, I cannot speak for 100 plus countries because I have not spoken to 100 plus regulatory groups. But I think everybody wants to be very aggressive while being careful with the safety profile. I think it is very different from what's happening on the treatment side of things. The treatments that are being tested as we speak in clinical control studies that you would give to people that are sick, because those people are very sick, in the ICU setting, the risk-reward profile is way different. Everybody is willing, the patients, the clinicians, the regulatory agency, to take more risk and to go faster with those. Some of those treatments, as you know, are already approved with the FDA for other indication like malaria, for one of these treatments that is being tested. And so we have years and years of safety data on that molecule to understand the risk benefit. And that's why I think the vaccine and the treatment need to be looked at differently. I think the FDA is very aggressive in their thinking while being careful so that we don't hurt anybody.
Yeah, it's actually having a thread, a very, very narrow needle
Yes.
in order to be able to balance those two
Yes.
considerations.
But the piece that is really amazing is the quality and the frequency of the dialogue with the agency.
Yeah.
It's very, very different than usual vaccine development. It's a real-time discussion.
Well, in fact, I mean, you've credited not only the FDA, but you've also credited CDC, NIH, BARDA, and other organizations. You've mentioned that this kind of collaboration is really unprecedented. Isn't that right?
Yes, I've been in this infectious disease business for 25 years. And so I've seen a few of those, outbreaks. I've never seen a global pandemic, unfortunately. So this is the first one.
Unfortunately.
Fortunately, sorry. But it has been really remarkable. We started designing the vaccine the day the sequence was online. If you look at previous outbreaks, sometimes it took months before the world knew the sequence. I have to credit the Chinese for that. They put the sequence online very, very quickly. Mm-hmm. And then within the US government, the agencies have been working together extremely tightly. Same thing, we have access to Dr. Tony Fauci, who has done a remarkable job for the country. Anytime we need to talk to him, the few times I've been in DC in the last few months, Tony has always made the time to talk to us with his team to make sure that we have access to all the resources of the NIH, CDC, BARDA has been remarkable. So I think we are lucky to have all those agencies with leaders who understand infectious disease inside out, know that we're at war against this virus, and wants to help industry because it's a partnership. We have a vaccine technology like mRNA. The government is helping us from funding standpoint, from access to science with Tony's team, and of course the FDA to help us navigate the regulatory pathway.
Yeah. So I'd like to come back to that partnership aspect in just a few minutes and talk about some of the economic considerations, because that's something that weighs on my mind. But I have one more question about the current situation with regard to the vaccine you're developing right now, and that's from Laura Sutcliffe. Her question is about the manufacturing capacity. Can you really scale up to produce the hundreds of millions or ultimately billions of doses that we're going to need to be able to treat the world?
So let me put a few caveats before I give you a bit of color. We don't know the dose yet. We will not know the dose until we have the immunogenicity data, which I say will be late spring, early summer. So the statements I'm gonna make are caveated by the dose, because as you can imagine, if we need 25 microgram, which is our low dose, or 250 microgram, you have a 10x factor right there in terms of course, how much mass of mRNA you can put out of a factory in terms of number of dose equivalent you'll be able to make. And so what we are doing now, and part of the discussion with BARDA is helping us on that front, is to scale up our manufacturing process so we can make bigger and bigger and bigger reaction, so that for every unit of time, we can make more mass of mRNA. The piece where we are lucky is if you look at the dose I mentioned, the 25 microgram of 100 microgram dose, those are per human, not per kilogram, like most of drugs are given per kilogram based on the weight of different patients. And if you think about it, a microgram is a millionth of a gram. So it's a very, very tiny quantity of material that you need for every different human being. So that's one of the things that we're lucky with vaccine, is we need a very small amount of mRNA for every dose we make. So what we're doing to scale up our manufacturing process, and I'll get to volume in a minute, is scaling up the process. So we started doing that with our own teams, with our own capital. As part of the BARDA discussions, there should be some help there if we get to a plan together to help us go faster and bigger on process scale up. And our engineers are working night and days and weekend, as you say, to just scale the process as fast as we can. The second piece we are doing is we've already placed orders for new equipment. Our plant in Massachusetts was overbuilt by design. We made it much bigger than what we needed, which we are super happy today that we did so. It was, of course, many millions of dollars that we invested to get this done, but we have basically several rooms inside the plants that are clean rooms. They are fully finished, but they have no equipment. They're just empty. And we had built them to help us scale the company. And so one of the things we have done with our head of manufacturing, after talking very quickly on a Sunday with our board, is to release some of our own capital to place orders for all the machines for all those empty rooms. Because of course, as you can imagine, it's months of lead time to get those equipments into the building. And then because we're going to make clinical material, it is very important that we validate and qualify those machines, that they are working properly using FDA guidelines and FDA law, so that we make high quality product, because again, safety is number one priority here. And the third thing we are doing is we are scaling up our teams. Today, our plant is working 2 shifts a day, 5 days a week. And we are hiring people as we speak. We have a lot of new employees joining us just in April already, and we have many more coming in the months to come, to go 24/7. We want to run the plant nonstop, because we know every additional gram of mRNA we'll make will literally make thousands of those available, if not hundreds of thousands when you do the math. And that's really, really important. And so where is that leading us? So we should be able, if all those things work according to plan, so again, there's a lot of caveats, scaling up the process, new equipment arriving, being validated, hiring people, training them. Again, this is a regulated business. We have to train people. It takes, many, many weeks. You don't train somebody to go into a clean suite, in 2 hours, and then they go make a medicine that you're going to inject in somebody's body. It's, again, very important to do things very well from a quality standpoint. And so our goal is to basically increase the output of a plant almost every month by doing those 3 different things in parallel and keep scaling up the process. And so we believe we should be able, if all those things work well, to get into the millions of doses per month in the short term, and in the dozens of millions of doses per month, more towards next year. And that's just with the current plant. We're also looking at potentially enabling other plants as we speak. Nothing has been finalized, but we're also looking into it to add capacity by just having more plants running in parallel. Mm-hmm. So how are we thinking about it? First, as we all know, we need to go on a risk-based approach, which is, medical workers needs to be vaccinated urgently because they are heroes in today's world. They are in the front line, getting exhausted, fighting the viruses. Because they're in hospital, they are getting infected all the time. So their body gets very high quantity of virus load, if that makes sense, in their body. And because they work those crazy hours, of course, the immune system gets weakened day after day, which is why you see them getting sick, which is very, very sad. And so the healthcare workers, the elderly, people that have comorbidity. Another piece that we are working very closely with is diagnostic company. As you know, we now have tests and the country is ramping up capacity very quickly to test using DNA or genetic type of test, to test is the virus present in the throat, in the nose of people. So that is a test to tell us, is the virus present right now? The test we need very quickly now is a serology test. I was actually on Monday talking to Senator Cassidy, who is a clinician, who is pushing really hard with a lot of people on the Hill. And Congressman Foster, if you can help there, that would be really wonderful, to help to have a federal registry for serology so that we can test in the months to come when the tests become available, and many manufacturers are working on them as we speak. I've been talking to 2 of them in the last few days, to take a test where you could take a blood draw of somebody and you can measure the antibodies in their blood. So you will be able to know, have they been infected or not? And as we have seen with other viruses, I want to be cautious because we're still learning a lot about this virus. But with other viruses, what we have seen is once you've been infected once, there is a possibility that you don't get reinfected easily or soon. And so as you can think about it, another population that we might want to focus on are people that have high comorbidity and are seronegative, meaning they have never been exposed to the virus. Those people are very, very high risk. And so you can start to think about a stratification of people around the country. We're basically working hand in hand with the government, especially with the CDC, is to decide, the first few million vials, who do you go vaccinate? And the month after, and as you get more and more vials done month by month, to just go on a risk-based approach to vaccinate as many people as you can. Which is also why I think having several manufacturers working on different vaccines not only helps on having more vaccines, having a chance to get to the finish line, that we have safety and immunogenicity, going back to your probability of technical success, Andrew, but also just to have more capacity online, because those are different technologies. Johnson & Johnson, as we know, is being helped also by BARDA. They have, a large manufacturing capacity. Their technology doesn't go as fast as our technology just because of how the science work. But the benefit they have is they already have manufacturing capacity while we are building ours, because the company Moderna is not commercial yet. So you just have those different players. And I think what BARDA is trying to do very well is to build a portfolio of different possibilities to make sure we have several vaccines that get to the finish line, because I think the world is going to need several vaccines.
So can I just stop you here for a minute to talk about diagnostics? Because as the former CEO of bioMérieux, a very successful French diagnostics company, you're deeply knowledgeable about the space of diagnostics. And I want to bring up an idea that was first proposed by European economist Matthias Duatrepont, University of Brussels, and his colleagues there. He pointed out that having a method for identifying people who are immune to COVID-19, that's actually part of the economic recovery. Because if we can tell who's immune and can get back to work, we can actually start restarting the economy with those individuals. So having a serological test to be able to identify COVID-19 antibodies is really important. And a couple of weeks ago, a group in Mount Sinai posted a paper on medRxiv that claimed that they have identified a serological test for this. how far away are we from being able to have an FDA-approved assay to be able to determine who is immune and be able to start getting people back to work?
Yes, that's an excellent question. So I think we are months away, and when I say months, I mean a few months away. So what I'm hoping is that by the summer of the early days and weeks of the summers, that we could have a test approved. Most, if not all, manufacturers of diagnostic tests that have immunoassay serology capabilities are working on them. If I'm correct, the NIH, I think, has been shipping to many of them antibodies, so they can use those antibodies to basically put into their systems. A lot of those companies, like the company I used to work for, bioMérieux, we used at the time, and this was 10 years ago, to have more than 60 or 70,000 machines around the world, many of them in the US, as you can imagine. Yeah. And so the amazing thing about this type of machine, and there are many manufacturers that have those big machines like Roche and Abbott and all those big companies, is as soon as you have the assay approved by the FDA, the next day, literally, you could have all those machines in all the big hospitals, all the big lab chains like LabCorp and Quest be available. And this is why Senator Cassidy is really trying to build this IT database where you could have this national database. Of course, that would be anonymous, but for the government to be able to know who is seropositive or not. The piece that we have to be careful because it's a new virus is we need to understand if you are seropositive, how long will it take for you to not transmit the virus anymore? Because what you want to be careful if is a seropositive is too quickly close to a seronegative, he or she might infect that person that has not been infected yet. And that's something really important to understand so that we do that correctly from a public health standpoint. But it is definitely the way to go. It will help put back people to work, which is of course really important. The economy is hurting and people and families are hurting around the world, as we see sadly in the news every day. No. And it will help, as I discussed as well, with the lack of availability of billions of vaccines, in the fall, next winter, next spring, so that you could focus on people that are at high risk and seronegative.
Yeah. Well, actually, along those lines, I guess in order to get that data, wouldn't you need human challenge clinical trials? And if so, what do you think the ethical considerations are going to be?
So there are many ways to get a vaccine to approval. You could run an efficacy study. There, the discussions that will have to happen is around the placebo group. But you could run an efficacy study given there is outbreak everywhere. I don't think it would be really hard to get an efficacy study up and running as a typical phase 3. The other piece that I know many academic groups, including the NIH, are working on is to develop an animal model where you could run a challenge model in animal, like the rabbit model I showed you on that slide earlier. Because there has been vaccine approved in this country, like in other countries in the world, where if you can show a phase 3 showing safety, so thousands of subjects enrolled where you can demonstrate safety, and you have an animal model that is considered by the right advisory board to the FDA as being relevant to what it will tell for humans, that you can have sometime vaccine approved using that method. The other one is the one you just mentioned, is to run a human study. I know there's been quite a number of papers published on the ethics of it, on how would you run it. Of course, there has been a few challenge study run in humans before. I can think of the flu. I can think of RSV, which is another respiratory virus that still does not have a vaccine today approved. And you can do that from an ethical standpoint if you have a treatment that is working safely. So I think having a treatment is a key consideration. Again, we as a vaccine sponsor cannot drive this process for all the ethical reasons you can understand. So we are following the space very closely. If an academic group or a clinical group in one of the leading hospitals in the country will develop such a model, working hand in hand with the FDA, once we have, I hope soon, a treatment approved so that if the vaccine at a certain dose does not work, we do not put the person at risk. But as you say, Andrew, there are many ethical considerations to be very carefully weighted. and this will have to be done through ethics committee with a lot of discussion and so on, because nobody at the agency or sponsors or of course clinicians in hospitals running those studies wants to hurt anybody, and so safety is very important.
Sure, of course. So I've got one more scientific question before we turn to the business side of things. We have a question from Jonathan Roberts. It seems your COVID-2 vaccine is based only on the S protein. Isn't there a risk that the virus could mutate to render your vaccine ineffective? Why not target other proteins instead of just S to reduce the risk of inefficacy against such mutations? And then a related question by Shan Shu is, do you think the full length of S protein is the right vaccine target? There have been accumulating papers suggesting the receptor binding domains of S protein might be more immunogenic.
Well, there's a lot of things to unpack here, but those are all good questions. So the spike S protein, as I said, the work we have done with the NIH on the Middle East respiratory syndrome, which is also corona, over the last couple of years, was very instructive. We tried a lot of different protein in that vaccine discovery effort. And every time we try different constructs and different combinations, because like I said about our CMV vaccine that has 6 mRNA, the number of mRNAs to code different proteins doesn't scare us. In our cancer vaccine, just to give you some reference, we code 34 antigen in each vaccine we give to every different human. And so the CMV vaccine has 6 mRNA. The EBV vaccine is Epstein-Barr virus. That's a virus causing mononucleosis, has 5 mRNA. So we just want to follow the science. The difference of cost of good or the difference of complexity in manufacturing is irrelevant. And so every time we try different combination for MERS, the spike S ended up being the most relevant, giving us, as you saw in the rabbit model, a really good performance that we and Dr. Fauci's team feels very good about. It's a full-length spike S. Unlike other technology, we don't have to do little piece of the protein. Sometimes with some other technology, that protein is just too big to be able to do successfully from a manufacturing standpoint. This is one of the biggest mRNA we've ever made to the clinic. It has 4,300 base pair. So think about it as a lot of zeros and ones on the message to code that for that very big protein. On the mutation question, that's an excellent question. So corona are mRNA viruses. As you know, there are 2 types of viruses. There are mRNA-based viruses like flu or corona, and DNA-based viruses like herpes viruses or HIV. The DNA virus, because DNA molecule is much more stable because of the double helix, and the H bond are very stable. They mutate very, very slowly. mRNA virus, because it's a single-strand nucleotic message, tend to mutate much more often, like the flu. We are monitoring very closely in our scientific team, and again, talking to the NIH almost daily, all the genetic sequences that are being posted online, and there are more and more as the testing is going on, as you can imagine. So we're looking at all of them and using bioinformatic algorithm to just compare the base pair of the entire sequence of all the protein of the virus. The virus has started slowly mutating, so it's still small. And to the best of my knowledge, as of yesterday, it has not mutated in the spike S protein. So the mutation that's happening outside the spike S, we believe for most of them are not relevant to the potential efficacy of a vaccine. If the spike S protein started to mutate, we'll have to look very carefully at the 3D structure of a protein to understand as the mutation happened to a part of a protein that's going to be seen by the immune system. And if that's the case, we need a new vaccine because we're going to lose efficacy. We might not lose all of the efficacy, we might lose some efficacy. Or it's in a part of the S protein where we believe from a scientific standpoint, there is low likelihood to impacting efficacy. If it was the worst case scenario, which is in a relevant region of the spike S protein, one of the amazing things about messenger RNA is how quickly you can go from sequence to shipping product.
Mhm.
As we've shown this time, which was the first time doing it, it took us 42 days from the sequence to shipping product that are ready to go inject into humans. I believe we can go faster. It is extremely rare that Homo sapiens does their best job the first time doing a task. And this is the first time we did an infectious disease vaccine under a clock watch like this. And so do I believe our team the next time around could go faster? I do. Mm-hmm. And so if the virus were to mutate in 3 months or 2 years or 5 years, we will have the ability very quickly to change the sequence. And as it is done for seasonal flu, we believe that agencies like the FDA will be comfortable with all the work done before. If the chemistry for the mRNA is the same, if the chemistry for the lipid is the same, which it will be, and the manufacturing process is the same, if it's just a few nucleotide sequence change, the agency has shown in the past, if there's a good scientific rationale, to be comfortable to have a product be shipped to humans.
Well, given that there are going to be potentially a need for new therapeutics, and there are a number of therapeutics being developed now, both vaccines and other kinds of drugs and medical devices, do you think it makes sense to have some kind of a nationally organized adaptive platform trial, kind of like what they did with breast cancer with I-SPY, or brain cancer with GBM Agile, or LungMap, an adaptive platform trial that allows you to test multiple drugs and devices at the same time, and to be able to get these various different therapeutics to patients on an ongoing basis. Is that even practical, you think?
Yes, so I don't think necessarily practical across the entire spectrum, because again, we give vaccine to healthy people and you give drugs to people who are sick or very sick. But in those sub-segments, yes, Andrew, I think it makes sense. And I'm sure, I don't work at the agency, but I'm sure the FDA, the way they will have the different manufacturers do the trial is very, very similar. If you look at the design of our phase 1, it is not different from the phase 1 that we've done for flu or for CMV, because at the end of the day, for a vaccine, question number 1 is it safe? Question number 2 is how much titer do you make? And question number 3 is your antibody neutralizing to the virus? Yeah. And so all the clinical trial sites who are used to do vaccine clinical trials are really used to the machine and to the protocols. The big CROs have all the database set up to do vaccine clinical trial because there are a lot going on across the world. Yeah. So I think that we have the ability to look at that and to basically share resources and share learnings.
Great. Well, so since we're running out of time, I want to turn now to the last topic, which is business considerations. Mm-hmm. In addition to focusing on patients, which is what is front and center for Moderna and other companies right now, you obviously have to respond to and report to investors, shareholders. And so one of the questions that I've been wondering for the last several years now is, how is this sustainable if we don't get government support to be able to provide the appropriate pricing to keep companies like Moderna in business? And I want to be very careful and respectful in terms of bringing up the topic now, because anytime you talk about pricing, particularly in the midst of a pandemic where people are dying, it's a very, very difficult thing to mix. Talking about profits and losses in the same breath as human life is frankly offensive to many people. But the problem is that if we don't do that, if we don't have the right economic model, we're not going to be able to get the therapeutics. So how worried are you about the economic situation, and what at Moderna is allowing you to address that in a way that will keep your shareholders happy?
Yes, so that's a very important question. And as you can imagine, we think about that a lot across all our portfolio. As you said, we have spent no time, not one meeting, thinking or talking about price. Our only focus now is how quickly can we get the clinical trial going? How quickly can we make products, scale up manufacturing? This is our 100% focus for the team. And the team is working extremely long days, 7 days a week. And now we have been doing that for 2 months. And it's going to be a long marathon. So let me tell you a bit how we think about it at a high level. The public-private partnership that BARDA enables in the US, which is a very unique setup from the US. I know several European countries that are trying to emulate a BARDA-like organization. And I'm sure after this pandemic, they will have to because they are way behind the ball. The US was really leading by having such an organization set up by the government. It's helping us to be able to take risk that will make no sense for us to take from a financial standpoint. Doing a clinical study of spending millions of dollars on a phase 2 preparation, which we are doing now, making phase 2 material, which we are doing now, is millions of dollars that if a phase 1 fails, will go to waste. We are making the phase 3 material now, as I told you as well. We're scaling up the process. All those costs will go to waste if a vaccine fails. And the vaccine can fail in phase 1, it can fail in phase 2, it can fail in phase 3. And so those private-public partnerships are critical because if you think about Moderna, the reason we were able to do the vaccine in 63 days from sequence to injecting the first human is because we have invested so far in the last 10 years more than $2 billion of capital. Our shareholders have not seen a return yet of a penny because we are still losing and investing in the business around $500 million a year right now to run the clinical studies, to keep investing in the science, to build a manufacturing plant and so on. And so what BARDA and the US government is enabling us to do is to invest at risk and not in sequence or not spend too much time thinking about should we do it or not, because what nobody knows now, because it's a new virus, is Corona SARS-CoV-2 going to be like the flu and stay with humans forever mutating, which might be down the road a commercial opportunity? Is it going to be like other respiratory virus, like it stays with us, mostly impact the elderly, and you might need every few years, we don't know yet because we need the clinical data and the clinical experience to know is it a once a year, once every 5 years, or whatever the science tells us kind of boost that you require to keep protections for the population at risk. And that's how we think about it. As we'll go down the road to get closer to launch, we'll have to be, of course, very thoughtful about pricing. We know this is a public health matter. So it is not at all the intent of a company, especially given we have a platform, we don't have one product, we have many products, to use a potential position where we are the first one to put a price that is not justified or that is not reasonable, if you want. So we're going to be extremely reasonable, and we work with the insurers, with the US governments to figure out the way. Clearly, we cannot give a vaccine away for free because no company in the world can give a product away for free because you can do that for so long. Given we lose money anyway, we have no revenues. If we give away the vaccine for free, the company will go bankrupt pretty quickly because we have to pay the salary of the employees, we have to pay raw materials, electricity and gas and so on to make the products, like any company in the world. But we believe that the vaccine can be made affordable. And I'm sure that working with the governments and the payers, a bit like has been done for the testing. If you think about it, the testing is free for the end consumer, for the patient. But the diagnostic companies are selling their product at a reasonable price, which is the same price they sell over product for HIV or other type of viruses. I don't know, maybe they are, but I'm not aware of any diagnostic companies that has put a very high price on their testing because it will not be appropriate given the urgency. But basically, either the government and or the private payers are paying those companies a fair price that is reasonable like other products. And I think we will have exactly the same approach. It will just be totally unjustified and not the right thing to do to put a premium price on such a vaccine given the need. And we will, I'm sure, be in good constructive dialogue with the authorities and with the payers to do what's right for the country.
Mm-hmm. Well, I mean, this is a really delicate subject, as you point out, because in the midst of a pandemic, when you start talking about, let's say, raising prices or having higher than average prices because you're spending a lot of your own resources and taking greater than average risks, that comes across as price gouging. On the other hand, during normal times when there isn't an outbreak, very few people want to even talk about vaccines because why bother? There's nothing to worry about. But we know for a fact that these vaccines are critical because these outbreaks happen all the time in different ways, different countries with different severity. So the question is there any rational approach that we can take more broadly to the portfolio of infectious diseases? Now, Moderna has actually got a portfolio of vaccine products that you could put together someday. Would it make sense to offer some kind of a combo vaccine that basically takes care of a list of diseases and where you charge some kind of subscription prices the way that they've done in Louisiana with Sovaldi and Harvoni, according to one of my economist colleagues, Rina Conti's proposals?
Yes, so I think there's a lot of things that we should be doing after we take care of this pandemic. Now the focus is really on this one. As I said, I've been in infectious disease for 25 years, and if I had a magic wand, what I would like the world to do on the other side of this pandemic is invest in a very large plant where we could potentially make, 50, 100, 150 million doses per month. Because think about it, if we had that plant existed in the US, we could have made 100 million doses in the first 30 days from a sequence of a virus. That would have been extraordinary, because you could have, in the first location like Seattle and New York, where you see the first cases, you could have done massive vaccination. The other piece to enable that needs to happen, and it's also investment. And I think, again, with BARDA, we're on the same wavelength, which is you want to enable the top 20 viruses to do the preclinical work, to learn about them, so that then you can go fast if one emerges, either the very same one or a mutation of that one. Think about our product. Why Moderna was so fast to the clinic? Because we spent 18 months working on MERS. I am not sure if we had not done any work on MERS, if we could have gone so fast. So I think there's two things to think about, is that there's still a lot of virus, like Zika has no vaccine approved, right. Like Marburg has no vaccine approved, Nipah, I mean, you can go on and on and on with a very long list of viruses that have very high mortality. I mean, last time I checked the Nipah data, and it's always pretty messy data because it's small data set in terms of the N, but it's like in the 60% plus mortality. Yeah. So just think about what we are seeing now with Corona, if it was very high mortality, like an Ebola type mortality. Yeah. And so I think that there's a list that is very well known to the CDC, to the WHO, of around 20 viruses that you ask any infectious disease expert, they will tell you they have a nightmare that those viruses are one mutation away in their genetic material to have a much higher infection rate and to create another pandemic that could be worse than the one we're living now, which is hard to imagine because the one we're living now is just horrific. And so I think those are the two things that the world needs to do, is to invest. And it's not a lot of money. I mean, you're talking $10, $20 million per vaccine preclinical. So it is not a lot of money at the scale of a country. Look at the economic destruction happening on a daily basis in the US, in Europe, and around the world. And so I think that is one thing that has to happen. And then a big project, a big infrastructure project that will be done for the country, where we would want a private-public partnership where we could use the plant for commercial products when there's not a pandemic, that could be potentially funded by the government for us to build, because today I don't have a capital to build that plant, because it will be in a billion plus type of kind of investment, but it's not $50 billion type of number, where we could have an agreement with the government where if there's a pandemic, we will basically have to stop what we do in that plant and just focus on the pandemic at hand to very quickly do product. Think about what could have happened in Ebola, in Zika, if that infrastructure existed. And it's one of the discussions I really want to have with BARDA and the different government agency and with Congress and with the Senate, because I think this is really doable. The technology exists today. Right. We will not do our job for the world if we don't do this. And if the next one comes, either a big outbreak a la Ebola or Zika, or a full global pandemic, that will both be irresponsible. The technology exists. And the way I think about it, and maybe it's a way to close there, governments spend billions of dollars every year on nuclear weapons they are hoping to never use. So what about spending a couple $1 billion to build plants, teams, scientific work and projects so that we equip ourselves so that this never happens again? We can do it. We just have to work together.
And in fact, we have $2.1 trillion that we should be able to take a small slice of that, as you say, a couple of $1 billion to build that capability. So hopefully our policymakers are listening now. So we're out of time. I want to just leave you with one question, perhaps. And while you're answering the question, perhaps I can get the audience to fill out the final poll about how they found this particular webinar. But the last question that I'd like to ask for you and to leave our listeners with is, can you give us any advice to those students and biotech entrepreneurs who are in the midst of their careers, starting out or looking for funding? Any advice to them about what they ought to be thinking about over the course of the next 6 to 12 months with their careers? Some business advice for them.
Yeah, so the next 6 to 12 months are going to be complicated on the business side because, as you know, a lot of clinical trials are being slowed down or delayed or paused. And so a lot of biotech companies are going to have a very hard time with funding because those delays are very hard to quantify. But I think we should stay very hopeful. This is a century of biology. We are learning every day more and more about how our body works at the fundamental molecular biology from a mechanistic standpoint. There are papers published every day by MIT, Harvard, and labs all around the planet that teach us and help us understand how the body works. I believe the acceleration of medical knowledge is currently exponential. And so I would really suggest to the young entrepreneurs and the scientists to keep at it, to not be worried about the complicated time we're going to go through in the next months and quarters. We're going to win this war against this virus. I'm convinced about it. And on the other side of this, there are going to be extraordinary opportunity to invest more in biology on the for-profit basis, on a not-for-profit basis, with more and more partnership. We can address many diseases. And it's what really gives me hope every day, which is if you think about what the biotech industry has done in the last 40 years since Genentech and Amgen started, those early teams could not have imagined what has happened today with the PD-L1, PD-1 checkpoint inhibitors in cancer, with all those amazing drugs. And so I have the same vision, is that the drugs that this industry is going to invent in this country in the next 10 to 20 years are going to be just amazing. And we need to keep at it, put our head down, do great science, collaborate, not only in the private sector, but with academia and with the government.
Well, on that very positive note, I want to thank you, Stéphane Bancel, and all of your colleagues at Moderna on behalf of everybody here at MIT and really everybody around the world for what you're doing for all of us. And we wish you Godspeed and hope that you're successful. And for all of you who are watching, thank you for joining us. The comments will be, and the video will be available online shortly. And please stay safe and healthy, and we will see you next time. Bye-bye.